Hi everyone! This video is about antipsychotic drugs. For this lecture, we'll be reviewing how neurotransmitters work, what antipsychotic drugs are, how they function, and examples of antipsychotic drugs. Antipsychotics are the main type of drug used to treat schizophrenia. They help reduce psychotic symptoms by altering brain chemistry. To better understand how antipsychotics work, let us first review neurotransmitter function. This is a schematic diagram of how neurotransmitters work. Shown in the diagram are two neurons. The end of the first neuron is the axon, and very near it is the dendrite or the beginning of the next neuron. The junction of these neurons is called the synapse. So, the neuron with the axon terminal is the presynaptic neuron, while the neuron with the dendrite terminal is the postsynaptic neuron. As you can see, there is a gap in the synapse. This is called the synaptic cleft. Neurotransmitters are molecules that are produced in the neurons from materials that are readily available. They are stored in vesicles. So this is a vesicle, and inside it are the neurotransmitters. Their function is to carry signals from one neuron to another or from a neuron to another cell, such as a muscle. I have drawn the neurotransmitters in different colors to symbolize the fact that there are different neurotransmitters. So far, 100 neurotransmitters have been identified. For instance, dopamine is not just a single type of neurotransmitter. There are different types of dopamine. On the surface of the dendrite, I have drawn structures that resemble small balls. These are the receptors for neurotransmitters. As you can see, they are also of different colors corresponding to the colors of the neurotransmitters. When an electrical impulse travels through a neuron, it starts a series of events. First, the vesicles move towards the surface of the neuron. Next, neurotransmitters are released. And finally, neurotransmitters bind with the receptors. As you can see in the illustration, neurotransmitters and receptors are specific to each other. Therefore, binding will not occur randomly. For instance, a dopamine 2 neurotransmitter can bind only with a dopamine 2 receptor. So isn't the human body amazing? It is so orderly. What happens to neurotransmitters after they have transmitted their signals? It can be any of the following. First, some neurotransmitters may be broken down or degraded by enzyme molecules. These enzymes are specific to neurotransmitters. For example, acetylcholine is degraded by acetylcholinesterase. Monoamine oxidase degrades both serotonin and dopamine, and in addition to monoamine oxidase, dopamine is degraded by catecholomethyltransferase. Some of the neurotransmitters may diffuse and may finally end up in the liver where they are metabolized. Some may end up in the kidneys wherein they are going to be excreted from the body. Some of the neurotransmitters, on the other hand, may go back to the presynaptic neuron through what is known as a reuptake system. This is some sort of recycling that happens. How do antipsychotics act? They act by blocking the receptors. As you can see in this illustration, the neurotransmitters have been released from the presynaptic neuron, but they cannot bind with the receptors because the antipsychotic medications have already occupied them. It is just like a game of musical chairs or trip to Jerusalem. You are the neurotransmitter and the chairs are the receptors. You lose the game because the chairs are already occupied by others. In this situation, the antipsychotic and neurotransmitter molecules compete for a limited number of receptors, thereby decreasing the binding of neurotransmitters to receptors. As a result, there is a decrease in the transmission of signals. Antipsychotics are also called psychotropics or neuroleptics. They are not curative, but they often decrease the intensity and severity of symptoms and therefore permit a person with schizophrenia to function in a supportive environment. There are two main types of antipsychotics. The first generation or typical antipsychotics were developed in the 1950s. These drugs decreased psychotic symptoms by blocking dopamine receptors. They also block acetylcholine and histamine receptors. The second generation or atypical antipsychotics, on the other hand, decrease psychotic symptoms by blocking serotonin receptors and to a limited, selective extent, dopamine receptors. What are the implications of these differences? It is thought that second-generation antipsychotics are more effective since they block both serotonin and dopamine. However, this has not really been proven. What has been proven already is that there are side effects that are more common in first-generation antipsychotics. These are extrapyramidal symptoms and anticholinergic side effects. Because of the lower incidence of these side effects, compliance is better for second-generation antipsychotics. These are examples of antipsychotics. The first-generation antipsychotics have been divided into two, low potency and high potency. For low potency, this is represented by chlorpromazine, which was the original antipsychotic developed in the 1950s, which changed the way clients with psychosis were treated. However, at present, this is no longer considered to be the drug of choice because it has a low potency and it is associated with a lot of side effects that tend to promote low compliance. Instead, high-potency first-generation antipsychotics may be used, which could be in the form of fluvenazine or haloperidol. Second-generation antipsychotics are actually more popular than the first-generation antipsychotics, represented by clozapine and risperidone. Clozapine, by the way, is the gold standard for treatment-resistant psychosis.